Okay, so sorry about the whole Zoom delay, but for this review session, we're gonna be talking about the second half of topic 5, so cell signaling and signal transduction. So like the RTK map pathway is gonna be found in the review from Tuesday, which is also on YouTube. So first, we're gonna be going over the survival pathway. So this one, the end result of this pathway is the survival of a cell. When it's not activated, programmed cell death is the result. And the pathway is really important in cancer biology research. And this pathway works with other pathways to help the cells survive. So that's an example of crosstalk. So first, the signal binds to RTKs, which causes them to dimerize, and then they cross-phosphorylate each other on the tyrosine. And then instead of those groups phosphating, adding a phosphate group to normally a protein like in the other pathways, it's going to be a lipid. And this lipid is called inositol phospholipid, if I'm pronouncing that correctly. And then the PI3 kinase binds to the phosphated tyrosines through its SH2 domain. And the SH2 domains always bind a phosphated tyrosine. Oh wait, can you just go?
Yeah.
Sorry.
Wait,
And then
did it go back?
no.
Okay, we
Okay.
go.
The inositol phospholipid is anchored in the membrane, and just again, it's not a protein, it's a lipid. Okay, now you can. So then PK1 binds to PIP3, which is the phosphorylated lipid anchored to the membrane through a pH domain. And then this activates the PK1, which then phosphorylates AKT, but the AKT isn't fully activated by just PK1 phosphorylating it. So when PK1 phosphorylates AKT, it like changes the conformation. It'll diffuse from the lipid anchored to the membrane, and it'll like reveal another amino acid that can be phosphorylated. So then while it's in the cytosol, PK2, which is floating in the cytosol, it's not anchored to the membrane, can phosphorylate AKT, and then it gets fully activated. Does that make sense? I could like clarify if anyone has any questions. Yeah.
So actually, no. Actually, on the problem sets, it's asking about mutations. Is it ever possible for PK2 to bind to AKT without PK1 first phosphorylating, or first binding it, or phosphorylating?
No, because it needs PK1 to, when PK1 binds or phosphorylates AKT, it like causes the conformational shape change that'll reveal like an amino acid that can be phosphorylated. And so if that doesn't happen, PK2 doesn't have that amino acid to phosphorylate, if that makes sense. Anything else about this slide? Okay. Okay, so then once AKT is fully activated, it binds to BAD, and that causes BAD to become inactive. So it's important to note that phosphorylation doesn't always necessarily mean activation. It just causes like a shape change, and then whether or not that shape change activates it or deactivates it depends on its function and in the pathway. So in this case, phosphorylating bad causes it to become inactive. So it causes a shape change, which causes it to release BCL2, which is the protein in red up there. And then BCL2 is free to bind to the mitochondria, which prevents cell death. So when BCL2 is still remain bound to bad, then there's nothing binding to the mitochondria preventing apoptosis. And then this is kind of just like a little summary page. So bad is the protein that allows for apoptosis when it's active, because when it's active, it's bound to BCL2. And then BCL2 is the protein that inhibits apoptosis when it's active, so when it's not bound to bad. And then there's just two scenarios here on the bottom. So when bad's active, bound to BCL2, BCL2 is inactive, apoptosis, and then when bad's inactive, it's not bound to BCL2, BCL2 is active, and there's inhibition of apoptosis. Okay, and then this is kind of just the same thing, but showing it again. Okay, and then here's just a summary of it. So survival signaling begins when a survival signal binds to the RTKs, causing dimerization and autophosphorylation. This allows the phospholipid cannot say this, inositol PI3 kinase to use its SH2 domain to bind to the RTK, thereby activating PI3 kinase. And then PI3 kinase phosphorylates the lipid that's anchored in the membrane, which causes it to go from PIP2 to PIP3. And then PIP3 serves as the binding site for PK1. And there's also another one. It also serves as the binding site for AKT as well. PK1 phosphorylates AKT, which causes AKT to dissociate from the PIP3 in the, that should say, I'll fix that before I release the slides, but it's anchored to the membrane. It's not in the cytosol. And then that allows AKT to diffuse in the cytosol and also reveals that amino acid that can be phosphorylated by PK2. And then active AKT can phosphorylate bad, which causes a conformational change, which releases the apoptosis inhibitory protein, which is BCL2. And then once BCL2 is active, it can bind to the mitochondria and inhibit apoptosis. And then here's just the entire thing together. So also, I don't know if it's on the problem set or not, but for example, if something says like PK1, there's a loss of function mutation in it, but then let's say there's like a gain of function mutation in AKT, then it wouldn't matter that there's a loss of function in PK1 because AKT is further downstream in the pathway, so that would lead to inhibition of apoptosis all the time. Does that make sense?
Because AKT is further downstream.
Yeah, so if AKT had a gain of function mutation, but let's say BAD also had a gain of function mutation, then it wouldn't matter that AKT does, because BAD in its active state is always inhibiting BCL2 from being active. So then it would always lead to apoptosis. That makes sense. Okay.
Okay. Now we're gonna talk about the JAK-STAT pathway. So we're first just gonna cover what RTKs are, and then we're gonna talk about discuss whether JAK-STAT is an RTK or not. So receptor tyrosine
Do you guys have a question? Sorry. Oh, okay.
Okay, so receptor tyrosine kinases, so the the receptor monomers, the O2 phosphorylate each other. That's how we kind of know it would be a receptor tyrosine kinase. Yeah, that's like the main point. In this, it is a signaling molecule that causes dimerization when it O2 phosphorylates. Just the main point here is that we are having the two monomers O2 phosphorylating each other. Okay, now we're just gonna discuss whether JAK-STAT is an RTK or not. And it's not. And the main reason it's not is because it's not a receptor. So it's a tyrosine kinase, but it's not a receptor. And the reason being is that the two receptor monomers, they do not autophosphorylate each other, but they have JAK subunits, which are proteins. The O2, so we have JAK1 and JAK2, and the O2 phosphorylate each other. And then after they're autophosphorylated, then they phosphorylate the receptors that they're bound to. And JAK itself is not also an RTK, it's just a protein subunit that is bound to another receptor. Does that kind of make sense so far? Okay. And like JAK literally just stands for just another kinase. That's what it is for. Yeah, you have something to say?
Oh, wait, can you go back? A quick thing. It mentions how the JAKs are subunits. So they're like, just to make it clear, they're separate from the RTKs themselves. Like the JAKs are not a domain, a part of the RTK. It's its own separate protein. So sometimes there's like questions asking if the RTKs in this pathway cross-phosphorylate each other, and it would be no, because the JAKs are what are actually performing it.
Yeah, so the JAKs are not part of the monomers, like the receptors. They are like two protein subunits that bind to that receptor, and they autophosphorylate each other. And then after they get phosphorylated, they then further phosphorylate the receptors themselves and cause the dimerization. Alrighty. So we're gonna start talking about the pathway itself. So we're gonna have JAK1 and JAK2 right here. They're gonna bind to like each receptor. And just to make it clear, we're gonna talk about the lactation signaling, so producing milk. And so we're gonna have after JAK1 and JAK2 bind to the receptors, we're gonna have the prolactin signal binding to the receptors, and this will cause the dimerization to happen. And after that occurs, we're gonna then have the JAKs phosphorylating each other. And after they phosphorylate one another, they're then gonna add a phosphate group to the receptor. And then after we get phosphorylation of that receptor, we're then gonna have the STAT5 binding to the receptors. And after they bind, so sorry, I just want to make sure I say it. They have an SH2 domain. So that they binds through it. And then after binding to the receptors, JAK1 and JAK2 are also going to phosphorylate STAT5s. And that phosphorylation of the STAT is going to cause a conformational change that is going to cause it to change shape and dissociate from the receptor. Does that make sense so far? Okay, and then after we have them dissociating, the two STATs are going to come together and form a dimer. And then they're going to be brought inside the nucleus through important, and they're going to act as a transcription factor. Okay.
So it's not correct to call the receptor an RTK, right?
No, because we said that RTKs, the two parts, so after they dimerize, the two parts of the receptor, they autophosphorylate each other. They don't have something from outside binding to it, like to the receptor, to then cause phosphorylation of it. Where like for the JAK-STAT, we have these JAK subunits, which are not part of the receptor. They come and they bind to the receptor after it dimerizes. And then they are gonna cross phosphorylate each other. So JAK1 is gonna phosphorylate JAK2 and JAK2 is gonna phosphorylate JAK1. And then after that happens, these are gonna phosphorylate each monomer of the dimerized receptor. And since JAK is not part of the receptor, that's why it's not an RTK. So it's not a tyrosine kinase receptor, it's just a tyrosine kinase. Okay. Is that clear? Okay. Alrighty, so just a quick summary. I'm gonna go over it again. We're gonna have a signal binding. In this case, since we're talking about the lactation, it's gonna be a prolactin signal, gonna bind to the prolactin receptors, and then after binding, that is gonna cause dimerization. And we're gonna have JAK1 and JAK2 cross phosphorylate each other, meaning that JAK1 will phosphorylate JAK2 and JAK2 is gonna phosphorylate JAK1. And then after they phosphorylate one another, you're gonna add a phosphate to the receptor itself, phosphorylating it. And after the receptor is phosphorylated, that causes like, not a conformational change, but that makes the STAT5 to bind to the receptor. We're gonna have two STAT5 molecules binding. And after they bind to the receptors, we're gonna have also JAK1 and JAK2. Each one of them is gonna phosphorylate one of the STATs. And after we get phosphorylating of STAT, it's gonna cause a conformational change, causing it to dissociate from the receptor. And then we're gonna have these two STAT molecule monomers coming together and they're gonna form a dimer. And that dimer of STAT5 is active, now in the active form, and it's gonna go inside the nucleus through importin. And it's gonna act as a transcription gene or transcription factor. Does that make sense? Does anyone have any question?
No one's
Okay.
said anything in the chat.
Yeah.
Oh, wait. I got kicked.
Oh, no.
I'll join again.
Okay. Alrighty. So now we're gonna talk about, this is just another thing together. You guys can practice like saying it to one another, to your friend, to your dog, to your cat, or yeah. Okay, we're gonna talk about G protein coupled receptors. Alrighty. So this is like a summary diagram, but we're gonna have another one too. Alrighty, so GPCRs are made of 7 transmembrane receptor, and they're bound to G proteins that is made of 3 subunits: alpha subunits, beta, and gamma subunits. And the alpha subunit is a GT base. That means it hydrolyzes GTP to GDP. Okay, so we have in the inactive state, we're gonna have the G alpha subunit of the GPCR bound to GDP. And then after, when we get a signal or a ligand binding to the G protein coupled receptor, then that is gonna cause activation for G alpha, and that happens, that G alpha is gonna let go of GDP and GTP is gonna diffuse in. And that will cause the activation of G alpha. And when G alpha is activated, it dissociates from the G beta and G gamma subunits complex. And it's important to know that the G beta and gamma subunits complex, they're always together, they're always bound, they don't dissociate like from each other like the alpha subunit does. So then now we have the G alpha could go and bind to another like effector, another enzyme, or it's gonna have like downstream effects in the cell. And then we can also now have gamma and beta subunits activated, and they could also go and act on another effector or an enzyme and have like a signal transduction cascade happening. So now if we then want to deactivate G alpha, it could happen just by G alpha inactivating itself since it's a GT base. So it's gonna hydrolyze GTP to GDP, but that's not really efficient. So we have RGS binding that so it's acting as a GAB. So it's a GAB analog, meaning it acts in the same way that GAB does. And what it does not hydrolyze. So it's not a GT base, so it does not hydrolyze GTP to GDP. And instead, it helps G alpha subunit does that faster. It just accelerates the process of G alpha subunit. And then after that happens, we're gonna get the G alpha and like the G beta, G gamma, all are gonna come together as like a one complex again, until we get another like ligand signal to bind to the receptor. Does that make sense so far? You guys have any questions?
I have a question, but I guess it's more about a different pathway, so I can save it for later. But like the quiz asked, what's a RAS activating protein? And the answer was SOS. So is that then different from something that's acting like,
Yeah, so RAS is from the same family of like RAN. So we have GAP and GIF help RAN does its work, right? They don't like, and correct me if I'm saying anything wrong. Like they don't do the hydrolyzation of like GTP to GDP itself. They just help that. Is RAN a protein? I think it is. Is it a protein? Yeah. Okay. Yeah. Okay. I just want to make sure. Yeah. So it just helps that protein like does its work faster.
It like stimulates the intrinsic catalytic ability. So it's not like giving the ability, it's just like helping it. So that makes sense. But
SOS also do that, or is it like something completely different?
so that's in the RTK RAS MAPK pathway.
Yeah.
right? So SOS binds RAS, which causes RAS to switch from a GDP bound state to a GTP bound state.
So then I guess SOS is more like, yeah.
Yes.
I'm sorry, I know that's
Wait.
not this. but-
No,
Yeah,
it's
just like the
fine.
gap. Okay. Does anyone in Zoom have any questions, or is everything clear?
Okay.
Okay, you guys can just stop us whenever you have a question. All right. So this is just another diagram like showing what happens. Okay, so here, this is like when G alpha is bound to GDP and it's getting deactivated. After this is the deactivated form of the G protein coupled receptor. So in this form, it would be waiting for a ligand or a signaling molecule to bind to the receptor. And after we get a signaling molecule binding, that's when we have activation of the G alpha subunit. And that happens by like GDP dissociating, and then we get GTP diffusing in. And after GTP diffuses in, then we're gonna have G alpha subunit dissociating from the receptor part, and it's activated. And then it's also gonna dissociate from the gamma and beta subcomplex, and they're also activated. And so now we have this complex that could go and act on another effector and have like signal transduction pathway happening. And we also have the alpha subunit can do the same thing. So it could have like a longer lasting effect on the cell than other types of receptors. Okay. Now, so we're gonna talk just about like some misconceptions. So we know that GIF promotes GDP to dissociate from GTP, and then we're gonna get GTP to diffuse in. So in that case, we have the GPCR acting as a GIF, because when we get a signal binding to that receptor, that causes GTP to dissociate from the alpha subunit, and we get GTP binding to the alpha subunit and activating it. Does that help? Are you confused? Do you have a question?
No.
Okay. And then we know that GAP, it binds to a GT base, and it helps accelerate the intrinsic activity of that GT base molecule. So it does not itself hydrolyze GTP to GDP, it just helps the molecule that is doing so like do it faster. So it just, I don't know, like increases the velocity of the process, I guess, the speed of that happening. And so this is just like some students would have like that misconception that saying that GAB itself hydrolyzes GTP to GDP, which is not the case. And RGS is also does not do this. So what RGS does is that it binds, it does bind, right? It binds to the G alpha subunit. And what it does, it just makes G alpha faster at doing its job, which is hydrolyzing GTP to GDP.
Yeah.
Is that clear? Okay.
But it's not actually.
Mhm.
Or I guess this is like more broad, right? But like in the JAK-STAT pathway, at least it's not actually GAP and GAP, right?
The JAK-STAT pathway,
Or
What the
GPCR.
GPCRs. No, it's not GAP or GIF. So we have the GPCR is what's acting as a GIF molecule. So it's called a GIF analog, means it does the same job as GIF does. But we don't have GIF in that pathway, no. And then we have the RGS molecule, which acts as GAP. So we don't have GAP, but this is a molecule that kind of does the same thing that GAP does. And just good to know that this like pathways in the cell are highly regulated, and it's kind of like to maintain equilibrium in the cell. So we have like activation and deactivation in the cell all the time. Yes, that's all we had for today.
Does anyone have any questions not related to the problem set? And then we'll do
The
problem
problem
set
set
questions
after,
after.
yeah.
No?
So I know that this before, but would SOS, well, we didn't talk about,
Okay.
so would SOS be a gas analog or a gas analog? Or neither, it could be neither, I don't even know.
It would
SOS
be
helps
a
gas.
gas
A gas?
Yeah.
analog.
It acts like RGS. right?
Yeah,
it'll be like
because,
it. Or
wait.
GPCR.
let me double check.
It converts GTP to GDP, right?
Yeah,
Yeah, so it acts as like
it's a GIF.
GIF. Yeah.
Because it's like performing the same like conformational change, so then it'll lose its affinity for GDP. GDP will diffuse out. And then we're always like assuming that there's enough GTP in the cell that it could just diffuse in. And then it becomes the activated RAS protein. So then it could phosphorylate MAP K3, which then MAP K2.
Can I see the question?
Yeah, does that make sense?
Yeah.
Okay.
Okay. So SOS helps
S-
GDP diffuse out and GTP diffuse in, right?
Yeah, because
Yeah, so it's okay.
SOS binds to RAS, changes its conformational shape, releases GDP. GTP diffuses in.
Yeah.
Actively.
Okay, does anyone, do we only have one person? Okay,
Yeah. Are there any other like
questions
Questions?
regarding
about the material?
Yeah,
It would be,
okay, I'm gonna stop the recording, just